This recording is for Unit 1.3, Patterns in Populations. Remember to take some notes on these as I go through it. Also, before we start officially, I want to talk a little bit about what a population is. This is a review of your 10th grade biology class, but I don't think it hurts. So population is a group of organisms of the same species. So this could be a group of squirrels, it could be this group of oak trees, but they're all the same species. Number two, those species also have to be in the same location. So we could have a population of squirrels here in New Ulm, and there could be a separate population of squirrels in St. Paul. There'd be two separate populations because they are in different locations. So we've got two important factors: same species in a population and same location. Okay, let's look a little more closely now at why scientists care about populations. The first thing they want to know is how big is the population? They have to come up with ways to measure it. So if we have a plant species, like the little blue plants in this picture, that we want to find the population of, this is fairly easy for scientists. They can go out and they can actually count those plants. That's called a census. Now, they don't go out and count all the plants in that area, but they choose little areas like our hula hoop experiment to count, and then they average it out for the whole area. So they can do a census pretty quickly and pretty easily. This gets a little trickier if you've got a mobile population or a moving population like bees or birds or mammals. So we can't really just sit and try to count them when they're moving around. Instead, scientists do something called mark recapture with these organisms. And mark recapture is kind of what it sounds like. So the scientists get some organisms, so they grab some bees, they catch them somehow, they put a mark on them of some sort. It could be a tag, it could be a radio collar. Lots of times with bees, they could just put a little spot of paint on them. They let them loose, and then they do a second capture of bees, and depending on how many bees they recapture from their original marked sample, that will give them an idea of how many bees are in the whole population. There's a whole equation that goes with this that we're not going to get into, but I do want you to know that there are different ways to actually measure populations. Another thing scientists are interested in when it comes to populations is the spacing within a population. We call this spacing the dispersion pattern, or how much room is between individuals of a population. In this first picture, what you're seeing is maple leaves with little tiny bumps on them that are actually something called galls. G-A-L-L-S. Now, galls can be caused by many things, but in this case, it's probably a little wasp that laid its eggs in the leaves, and the little wasp eggs are developing into larvae, causing this swelling within the plant, because the plant's trying to protect itself from these larvae. And you'll notice that there are little clumps of galls on the various leaves. So this is a special type of spacing or dispersion pattern that we call clumped. Easy to remember. So that is a clumped dispersion pattern. Other organisms that might show clumped dispersion patterns might be like sea stars on the edge of a cliff that are all clumped together in one area because of the tide coming up and down. All right, then we have another type of population dispersion pattern that you can see in this picture right here with these birds sitting on the wire. They look like they're not clumped as much as the galls are. In fact, they're pretty evenly spaced across this wire. So we call this dispersion pattern uniform because they have approximately a uniform distance between each one. They're each giving each other their own space. A specific type of bird that does this is penguins when they're nesting, for example, in Antarctica. And so their emperor penguin nests there, and it will give the other emperor penguins space so that they're not all nesting right next to each other. There's pretty much uniform amount of space between each one. And then this last picture right here is, I think, flamingos migrating. And you can see there's not really clumps of flamingos, and some are spaced farther apart than others. So it's not clumped, It's not uniform. We call this dispersion pattern random. Again, all of these names really make sense. They really describe what's going on with that dispersion pattern. All right, the topic of populations that we're going to spend a bit of time on in class is demographics. And you can see the definition for demographics right here. It's the study of the vital statistics that affect population size. Those vital statistics are births, deaths, immigration, and emigration. So there are 4 vital statistics. They each do a different thing, of course, to the population size. Births and immigration increase population size, immigration being new organisms coming in. Deaths and emigration decrease population size, emigration being organisms moving out of the population. So one way we can look at all of these births and deaths and immigration and emigration and track what happens over time is something called a life table. So a life table summarizes the survival pattern of a population. So it looks at all the births and deaths and immigration and emigration and puts them into one table to summarize what is happening to that population. When are there a lot of births? When are there a lot of deaths? When do we see immigration, emigration, and how are they all related? In life tables, how you organize the data is by something called a cohort. And a cohort is a group of individuals of the same age that's tracked from birth to death. And once you can track that cohort, you can really start to see some patterns in those vital statistics. Now this is kind of vague right now in just this description. I'm guessing you don't have a real great idea of what a life table is. So we're actually going to create life tables in class with data that you collect. And so your assignment for this video is not to fill out a feedback form, but to collect data for a life table. And how you're going to do this is shown here. This first chart is looking for male lifespans for Brown County. So you are going to create one chart just like this with male lifespans for Brown County. And I'll talk in a minute how you're going to get those male lifespans. But you can see by the first column here, we've got number indicated. And let's see how many numbers of males we need. Each number represents one individual. And we've got 8 rows for individuals. So we're going to come up with 8 males that lived in Brown County that have passed away and how long they lived. So if you've got individual number 1 you find, and his name is Joe Smith, and he died when he was 81 years old, his length of life is 81. Then you find another one. His name is John Doe, and he lived for 56 years, so his length of life from the day he died was 56 years. So you're going to find 8 males. Now you're also going to do this with females as well. And 8 females is what you're going to be looking for. Same data, name and length of life, but for females as well as males. So when you're done and prepared for class, for doing your homework, you're going to have 2 data tables like this. One for males, one for females. Now you're probably wondering, or maybe have an idea, where am I going to get this lifespan information to fill out my 2 tables? Well, the most common place we go to find length of life data is an obituary. We're going to go to the New Ulm Journal obituary because we're looking for Brown County data. The website is right up here, so you can put that into your computer. It'll take you to a page that looks kind of like this, and you'll see the information you need right there. So we've got our first individual listed here, Gladys Abraham. She died when she was 90 years old. So we would put her into our female table, name Gladys Abraham, age 90. Down below her you can see there's another female, Louise Garner, and she died when she was 74. So she'd be individual number 2 in the female data table. Put down her name, Louise Garner, and death at age 74. So you're going to keep filling out your male and your female tables with the information you find. Now the other thing, or data, the obituary gives you is the date of death. And I do want to separate your data collection into different dates so that people in the class aren't using the same people. I don't want everybody in the class to have Gladys Abraham on their life table data, because that's going to mess up our overall survivorship curve that we do this with this when we're all done. So what I've done is I've assigned to each a different time period for which you're going to be using. And each student will then go to that time period on the obituary site and find the males and females that died during that month. So for example, Rachel is going to go to August of 2013 on the New Ulm Journal obituary page and look through all the individuals that died August of 2013, choose 8 males and 8 females to put those into her 2 data tables. If there aren't 8 males and 8 females that died during that month, do your best. Get as many as you can. If there are more, you can stop at 8. So stop at 8 males, stop at 8 females. We don't need more than that. Now, how do you find your time period? On the obituary page, there is a search feature. So you'll be able to find where it says search on the obituary page, and then you can put in the dates from what month to what month you're searching for. All right, so again, your homework for this particular video is to fill out your 2 data tables with all of the information on 8 males and 8 females and bring them to class so that we're ready to go to make a life table.